Imagine two identical twins. Both have similar genes. Both grew up in the same environment. Yet only one develops systemic lupus erythematosus. If genes are responsible, why doesn't every genetically susceptible individual develop SLE? If environmental triggers are responsible, why doesn't everyone exposed to sunlight or viral infections develop lupus? The answer lies in the interaction between genetics, environmental triggers, hormonal influences, and immune dysregulation. Think of SLE as a 5-step disease. 1. Genetic susceptibility. 2. Environmental trigger. 3. Immune system loses self-tolerance. 4. Autoantibodies are produced. 5. Immune complexes damage organs. This simple framework helps explain almost every clinical feature of SLE. Step 1. Genetic susceptibility. Not everyone develops lupus after exposure to triggers. A genetic predisposition is essential. Several genes increase susceptibility, including those involved in immune regulation, interferon signaling, antigen presentation, apoptosis, and complement pathways. Important associations include HLA-DR2, HLA-DR3, IRF5, STAT4, TREX1, PTPN22. Complement deficiencies, especially C1q, C2, and C4. Remember, genetics loads the gun. Environment pulls the trigger. Step 2. Hormonal influence. SLE predominantly affects women during their reproductive years. Female to male ratio, approximately 9:1. Possible reasons include X chromosome gene dosage, estrogen effects on immune function, enhanced B-cell survival, increased antibody production. Interestingly, children and elderly patients have a much smaller gender difference, supporting the influence of hormonal and genetic factors together. Step 3. Environmental triggers. A genetically susceptible individual usually requires an environmental trigger before the disease becomes clinically apparent. Ever wonder why sunlight can trigger a painful flare-up for someone with lupus? It isn't just a sunburn, it's a biological chain reaction. Here is the sun-to-flare breakdown. UV exposure, ultraviolet rays hit the skin. Cell damage, this causes injury to healthy skin cells. Apoptosis, the body triggers programmed cell death to clean up the mess. The leak, as these cells break down, they release hidden nuclear antigens, pieces of your own DNA or proteins, into the body. The immune system sees these released particles as foreign invaders and attacks. This internal fire causes the malar rash on the face, photosensitivity, and full-body disease flares. In short, the sun causes your body to misidentify its own broken cells as an enemy. Viral infections, especially Epstein-Barr virus. Epstein-Barr virus stimulates B cells, and molecular mimicry may contribute to autoimmunity. Cigarette smoking. Smoking increases oxidative stress, enhances inflammation, increases disease severity, reduces response to therapy. Drugs. Drug-induced lupus is associated with medications such as hydralazine, procainamide, isoniazid, minocycline, certain anti-TNF agents. Unlike idiopathic SLE, drug-induced lupus usually improves after withdrawal of the offending drug. Step 4. Loss of self-tolerance. This is the most important event. Normally, the immune system distinguishes self from non-self. In SLE, this recognition fails. The immune system begins attacking DNA, RNA, histones, nuclear proteins, cell membranes. Essentially, the immune system mistakes the body's own tissues for foreign invaders. Step 5. Defective clearance of apoptotic cells. Every day, billions of cells undergo programmed cell death, that is apoptosis. Normally, dead cells are rapidly removed by macrophages. In lupus, clearance is defective. Dead cells accumulate. Their nuclei rupture. Large amounts of DNA and nuclear proteins are released into the circulation. The immune system interprets these exposed nuclear components as foreign antigens. Step 6. Dendritic cell activation. These nuclear antigens are captured by plasmacytoid dendritic cells. These cells produce large amounts of type 1 interferons, especially interferon alpha. Interferon alpha acts as an amplifier of autoimmunity by activating multiple immune pathways and promoting further B-cell and T-cell activation. Step 7. B-cell hyperactivity. Activated B cells differentiate into plasma cells. These plasma cells begin producing numerous autoantibodies. Important antibodies to know: ANA, anti-nuclear antibodies; anti-dsDNA, anti-double-stranded DNA; anti-SM, anti-Smith antibodies; anti-RO, also known as SSA; anti-LA, also known as SSB; anti-phospholipid antibodies. We will study each antibody in detail in a later chapter. Step 8. NETosis. This is one of the most fascinating concepts in modern immunology. What they are: neutrophils defend the body by releasing web-like structures made of DNA and antimicrobial proteins. What they are called: NETs, or neutrophil extracellular traps. Normally, NETs trap bacteria and fungi. In SLE, NET formation is excessive and NET clearance is impaired. Step 9. Autoantibody formation. Autoantibodies bind to self-antigens. How immune complexes form. Example 1. DNA plus anti-dsDNA antibody forms an immune complex. Example 2. Smith antigen plus anti-SM antibody forms an immune complex. Note: these circulating immune complexes become the major drivers of tissue injury. Step 10. Type 3 hypersensitivity. This is the hallmark immunological mechanism of SLE. Immune complexes circulate through the bloodstream. Where immune complexes deposit: in kidneys, skin, joints, blood vessels, pleura, pericardium, brain. What happens next? These deposits activate complement. Complement activation recruits inflammatory cells. The final result will be tissue injury. Complement consumption. Complement proteins are consumed during ongoing immune complex activation. What happens in active SLE? Decreased C3, decreased C4, decreased CH50. Key takeaway: low complement levels often indicate active disease and are especially useful when interpreted alongside clinical findings and anti-dsDNA levels. Organ damage. Immune complexes accumulate in different organs: kidney, lupus nephritis, proteinuria, hematuria, and renal failure; skin, malar rash, photosensitivity, and discoid lesions; joints, non-erosive arthritis and arthralgia; heart, pericarditis, myocarditis, and Liebman-Sachs endocarditis; lungs, pleuritis and pleural effusion; brain, seizures, psychosis, and stroke; blood, hemolytic anemia, leukopenia, lymphopenia, and thrombocytopenia. Now that we've understood why the immune system turns against the body, the next question naturally follows. What does this immune attack look like in a real patient? Why does one patient present with a butterfly rash, another with arthritis, another with kidney failure, and another with seizures? In the next chapter, we'll study the clinical manifestations of SLE, moving from head to toe and correlating every symptom with the underlying immunopathology.